Hello biology students, and welcome to video 7 of the DNA unit. So in the last video, we looked a little bit at translation, but in this video we're going to be looking at the molecular steps of translation. So in other words, how does the ribosome read the RNA, and how does the ribosome know what amino acid to add depending on what codon we have? So just like transcription, there are going to be 3 steps of translation: initiation, elongation, and termination. So those names should sound really familiar to you because they're the exact same names the 3 steps of transcription had. So let's look at these steps in a little bit more detail. Okay, so the first thing to know about translation is that when I have an RNA message, not all of that RNA gets translated into a protein. Just like we had a promoter and terminator sequence that tells us where to start and stop transcription, we're also going to have a sequence called the start and stop codon that we saw in the last video that tells us what part of the RNA to actually make into protein. So for example, if this pink molecule here were my RNA, I might the start codon might be right here. So this is where my AUG might be. And then maybe my stop is over here. So maybe I have a UAA right here. Okay, so not the whole thing gets translated into a protein. Okay, so like I previously mentioned, the first step is initiation. Okay, and what's going to happen? I don't know if you remember, but the ribosome is actually made up of 2 subunits. There are 2 pieces to the ribosome. So the first thing that happens is my mRNA binds to the small part of the ribosome. So you can see the pink RNA here has bound to the small part of the ribosome here. Then my first tRNA comes in. And remember, tRNA is the RNA that brings amino acids to the ribosome. And the way this works, each tRNA, if you remember, has an anticodon on the bottom. That's this UAC in green here. And then it has an amino acid attachment site here. And it turns out that every tRNA that has a UAC as an anticodon always carries a methionine. So conveniently, UAC, that anticodon, is complementary to our start codon AUG, which means every time we have an AUG, the tRNA with the UAC anticodon is going to bond to it, and that tRNA is always going to be carrying our first amino acid, which is a methionine, which is good because that's what the codon chart tells us lines up with AUG. So after this happens, the other half of the ribosome comes in, the large ribosomal subunit. And there are 2 slots on this large subunit. There's what we call the P site, which is the slot right here, and that's what holds the growing polypeptide chain. And then the other site, the A site, is right now it's empty. That's where the next tRNA is going to come in with its amino acid. Okay, so that was initiation. So our next step is going to be elongation. And just like in transcription, we're going to elongate, or here we're going to build the growing polypeptide chain, or the growing protein. So elongation adds amino acids to the polypeptide chains, and there are 3 steps to elongation. There's codon recognition, peptide bond formation, and then translocation. So let's look at those now. Okay, so the first step of elongation is going to be codon recognition. And this is when the anticodon on the incoming tRNA is going to recognize and complementary pair with the mRNA codon that's currently in the A site. You can see here, whatever these 3 bases in the mRNA are right here, they're going to pair up with the 3 bases in the anticodon, allowing this tRNA to bind in this A site, the spot that was empty in the ribosome. The next step is peptide bond formation. Okay, so this is when the ribosome makes a peptide bond. And remember, peptide bonds are the bonds between amino acids. So it's going to attach the polypeptide chain that's in the P site. It's going to bond it with the other amino acid that's currently in the A site. So if we look at our picture here, peptide bond formation, didn't mean to do that. If we look at our peptide bond formation picture here, you can see that the amino acid chain that was attached to this tRNA gets bonded to the amino acid right here. So now if you look, it's all bonded together in a growing polypeptide chain. The last step in elongation is translocation, and this is where the empty tRNA leaves, and then the one that has the growing chain on it moves over to the next spot. So you can see now it's in this spot here. So in other words, everything shifts to the left. And this frees up a new spot for our next tRNA to come in. Okay. Then finally, the last step of translation is termination. So elongation continues until we reach a stop codon. And basically, stop codons do not code for amino acids. So once I get here, there's going to be no tRNA that will have an anticodon that will bond with my stop codon. So this tells the ribosome that we're done translating, allowing the mRNA to leave, the protein to leave, the ribosome to split up, etc., etc. So you might be wondering, well, now there are some tRNAs that have dropped off their amino acids, and now they're empty. They aren't holding an amino acid. So what happens next? So those tRNAs that don't have an amino acid, each tRNA has a specific enzyme that will add the correct amino acid back to it so it can continue doing its job for the next mRNA that has to be translated. Okay, so the last picture I want to show you here, not the last one, the next picture I want to show you here is this picture down here is an excellent representation of termination, because we haven't seen a termination piece yet. So once you see the stop codon, which we assume are these three nucleotides right here, they don't code for a tRNA. So basically no new tRNA comes in, which is the signal that the polypeptide should leave, this tRNA should leave, the mRNA should leave, and the ribosome should break down into its subunits. Okay. So now we can summarize the entire protein synthesis process. So we know that DNA gives us RNA in transcription, and RNA is made into protein in translation. So the way this works is basically the sequence of nucleotides, or the sequence of codons in DNA, dictates the sequence of nucleotides in RNA, and that, through translation, dictates the order of amino acids in a protein. And this happens via first the transcription of mRNA. Then remember, I process the RNA, I cut out the introns, and I add a cap and a tail. tRNAs pick up their appropriate amino acids, and then I start building a protein, I do elongation, and then finally I stop building my protein. So we look at this whole process in one more picture to make sure we're okay. After I do transcription, mRNA leaves the nucleus. It's going to bind to my ribosome. Each tRNA anticodon is going to bond to the appropriate codon, bringing in the correct amino acid. I'll form peptide bonds between these amino acids. The empty tRNA leaves. The next tRNA comes in. I form another peptide bond, etc., 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 until I am left with my whole peptide chain and a stop codon where there's no tRNA will come in, signaling that the process is done. So that's it for video 7, and I'll see you in video 8.